everyone. So I'm going to be doing the intro today since I've known Trevor for a few years. So almost 12 years ago this week, I think that Trevor arrived at the University of Toronto to start his graduate degrees. He came to us from Laurentian University, where he had studied chemistry and done research in chemistry and forensic science. And he came to Toronto in 2007 and joined my research group, which was in its early days in the first few months. And I was really lucky to have him as one of the founding members of the research group. So during his time at U of T, during his PhD, Trevor worked on a lot of interesting projects. He took the lead on some analysis of alkylamines in our group. And he also did a lot of interesting and creative work looking at HONO and its surface atmosphere exchange, both through field measurements and lab measurements as well. And in recognition of the excellent work he did during his PhD, he was awarded a Banting Fellowship that started off his time at Boreal University in Newfoundland, where he did a lot of interesting field work focused on biogeochemistry. Spent a few years there and recently moved back to Toronto, where he is working at York University, and he's officially starting a position as an assistant professor in the summer. So please join me in welcoming him back to Toronto.
Thanks, Jen. Thanks, everybody. It's great to see so many familiar faces here, so many Murphy Group members as well. So you guys will be familiar with a lot of what I'm going to talk about today. And for the rest of you, I'm going to be sharing some of the work that I did during my postdoc, some nitty-gritty analytical work, looking at atmosphere-biosphere exchange of reactive nitrogen, and some of my new forays into reactive nitrogen chemistry in indoor atmospheres. So the new position I'll be taking up in the summer is really focused on analytical environmental chemistry. And so sort of the theme that I have for the talk today is to show sort of where that analytical work comes into making really robust measurements that we can trust when we go to do different types of work, both outdoors and indoors, in particular where we have very trace levels of the analytes that we're trying to measure. And so just to give everybody sort of a big picture about why I like to work on reactive nitrogen chemistry, this is sort of it. This is a summary that was done looking at global tipping points published by Rockstrom et al. Back in 2009, where they looked at a variety of different parameters that could be perturbed in the global system by human activities that could have potential knock-on effects in terms of positive feedback loops or tipping points that once passed put us into a world in which we don't really know the consequences of our actions. So things on here that we have studied a lot are things like climate change, where we know we've gone past certain tipping points. We live in a world with a different climate, and understanding the impacts of that is sort of what a lot of us are working on. And other things that are included in here are things like nutrient cycles, nitrogen and phosphorus. I can't see all of the nitrogen here, but that is totally off scale, and things like biodiversity loss, amongst others. When we perturb global systems, we wind up creating these unknown worlds that we live in. And reactive nitrogen in particular is just a consequence of feeding humanity. We have a growing global population. We need food and energy to grow that food, which means we require fertilizer and fuel. And that means we perturbed our global reactive nitrogen system in response to that. So as human populations have grown over time, the consumption of nitrogen on a global scale has increased in lockstep with that. And it's been growing exponentially as different countries start to consume more meat. All those animals have to eat vegetation, which is grown by commercial fertilizer production, and that has a knock-on consequence as well. So we have this global perturbation of nutrients, especially the nitrogen cycle, where the present-day reactive nitrogen cycle, that's everything that's nitrogen in the atmosphere, except for that dinitrogen and N2O, nitrous oxide, is really anomalous in Earth's history. And so the consequences of that are something that we're going to have to live with and try to understand in greater detail. In order to do that, we start to learn more and more about different reactive nitrogen species in the global environment. When we find new species that may have interesting consequences, we tend to find out about them with the use of new analytical techniques. And validating those techniques is what we need in order to have reliable measurements so that we can make informed decisions. So if we want to be globally responsible in understanding where nitrogen is coming from, what's happening to it, and its consequences, we need to have good measurements to do that. And this is just sort of another depiction of that. Humans fix reactive nitrogen at a similar rate to natural processes. So conversion of dinitrogen into reactive nitrogen, split pretty evenly between the biosphere and humans. And about half to almost all of that can be denitrified on an annual basis as well. So we could be increasing reactive nitrogen significantly on a global scale by up to 200 teragrams of nitrogen per year, or as little as 20 teragrams per nitrogen per year. In either case, we are dramatically increasing the global burden of reactive nitrogen on an annual basis every single year. So we perturb that by burning fossil fuels or using fertilizers. We emit these into the environment, whether it's the atmosphere, terrestrial or marine environments, and we can affect the cycling of reactive nitrogen within those and the transport between those two processes. And that sets off something we call the reactive nitrogen cascade. We put this new nitrogen into any of these reservoirs. We can affect the processes that go on between them. And that's where we have our large-scale impacts on global processes. But it's the linkages in the species connecting these different terrestrial, atmospheric, and aquatic reservoirs that are actually transporting reactive nitrogen around the world, and being able to make good quantitative measurements of the species that link those different reservoirs are the critical places where we need to be able to quantify the rates of chemical cycling. And so this is sort of a lead up to the work I did during my postdoc, where I was trying to quantify reactive nitrogen cycling in a boreal forest environment. So this took place out at Memorial University at a place called the Newfoundland and Labrador Boreal Ecosystem Latitudinal Transect, or the NL Belt. This latitudinal transect is set up to look at carbon storage in the boreal forest, particularly in soil organic matter as a carbon reservoir. And we wanted to understand how that carbon would be preserved under a changing climate in the future. And so the experimental design here is to trade space for time by setting up these forested watershed sites that span the west coast of Newfoundland into southern Labrador. We have 5 degrees latitude across the transect, and that comes with a trade-off of about 5 degrees Celsius mean annual temperature difference between the south and the northern sites. And so this is a natural experiment where we can perturb or have essentially a proxy for climate change without actually having to experience it directly. And so what the group had found out before I joined it was that they sort of had stable carbon stocks in the soil, and they didn't really understand why that was. So in a warmer environment, they saw higher cycling, but they didn't see a net loss of carbon from the soils. And what immediately stood out to me was, well, you guys are focusing only on carbon, and the coupled nitrogen processes that might be going on in this system could help us try and understand this better, and it could help explain what's going on. And in particular, there was no measurements of what was coming in from the atmosphere. That was my background specialty. And so I made it my mission as part of the team here to bring in an atmospheric reactive nitrogen subgroup to the NL Belt to quantify atmospheric reactive nitrogen inputs across the transect. And we figured that we had to do this because when we went to look at the outputs from global atmospheric chemical transport models, the reactive nitrogen deposition that we might try to derive from those model outputs were very uncertain. We were essentially sitting across this transect in gradient zones, so orders of magnitude difference in estimated deposition values out there, because most of the models were being driven by extrapolation from measurements made further inland on the continent, or sporadic measurements made on the island where these two stars are located. And so we had very uncertain inputs going on there. So we had this need for quantitative reactive nitrogen measurements, and we suspected that we'd be in very trace levels. So that required a lot of analytical work, which I love a challenge, especially when it comes to measuring trace reactive nitrogen compounds. And so I took that on. So this wasn't done in isolation. I had a subgroup within this larger NL Belt group that looked at all the atmospheric reactive nitrogen chemistry coming in. So all the work I'm going to be talking about today comes from a variety of students. A lot came from Brian Place, who's actually an alumni of U of T from the chemistry program, who did his master's with me. But a lot of work has also been done by Courtney Laprise, who is an undergraduate student, who did her undergraduate thesis with me. And some of the work I'm going to be talking about today actually comes from Rachel Hems's summer project when she was out in Newfoundland to do some work out there as well. But this is all in context with the bigger picture. So biogeochemical studies require all sorts of different experts. We had foresters, microbial ecologists, hydrologists, terrestrial and aquatic biogeochemists, all contributing to this project. So I'm going to be talking about what we found today. But as we work up this data, it's really going into this bigger context that's driven by this giant working group. So our job at NL Belt then was to capture as many reactive nitrogen inputs as possible, speciate them, and quantify them. We had seed funds to begin the monitoring of dry deposition. We did well with that pilot project, and I was able to then use my Banting funding to leverage a long-term monitoring project that was fully funded by the government of Newfoundland and Labrador to have a full monitoring program over 4 years to capture wet and dry deposition of reactive nitrogen coming into these ecosystems. And so the plan was we want to get dry deposition, wet deposition, decouple what's coming from long-range transport and local emissions, find out what effects that has on the local productivity, but also its linkages to the terrestrial nitrogen cycle and the aquatic systems. This is challenging in and of itself to begin with. We add on top of this the fact that we have analytical challenges, that there are no power drops in any of these sites. We have to do everything without power or find ways to bring power to them ourselves. And we can only get there once a month when we can actually access the sites because they are located in remote regions. That means that we have to be able to have our samples collecting automatically for all the entire time between those collections. And so this gives us a big challenge. This is just an example of how we approach those types of challenges. When we go to collect atmospheric gases, we did it by passive sampling. So we use the natural fact that gases diffuse in the atmosphere and they can diffuse towards a surface that we intentionally set up the chemistry of so that they get collected on that surface. And we can then quantitatively sample those gases as a function of time, integrate over a month, and back out the concentrations that were present at the sites. And so this is just an example of how we set up our gas collections. So we do a bunch of nested replicates within a site to get statistical viability of our different sample collections, collect a variety of different reactive nitrogen compounds, and then we try to achieve 3 things: sensitivity, selectivity, and reproducibility. This is challenging to maintain over a long term. We're doing monthly collections. This means we're making 6 to 7 trips per year. Each one of those is 7 to 12 days in duration. To get across that whole transect, you're driving 2,500 to 3,300 kilometers per trip, and you're bringing back 250 samples per trip that you have to analyze. So a lot of cases, we're going out into the field. Here's our passive samplers. We're climbing up into trees with these tall ladders, getting our samplers mounted on them, pulling down the ones that have collected, taking them back to a hotel room, trying to turn it into effectively a clean lab to do our extractions without contaminating anything, and then repeating the same thing the next day after driving 6 or 7 hours. And sometimes taking a ferry and getting blown in by wind and all kinds of stuff like that. So the QA/QC that we have to derive for this can be quite challenging. And so on top of that, once we get back to the lab, we have to have analytical methods that are capable of quantifying those. Luckily, we have a good team. There was a lot of funding available to help get students out into the field, especially the Northern Scientific and Training Program, in order to get students like Katie and Brian out to the field to help me collect samples and do all the sample processing in the field. Otherwise, this type of work isn't possible unless you have enough hands to do it. And so just to give you an example of a deep dive into the analytical chemistry here, I'm going to talk a little bit about one of our target compounds. This is part of Brian's thesis, was to sample nitric acid quantitatively in these remote regions. So when we have anthropogenic or natural sources of nitric oxides into the atmosphere, we can form nitric acid at both during the day and at night that can get transported downwind, either as nitric acid gas or as ammonium nitrate aerosol, which can be deposited into systems by dry deposition or wet deposition. Dry deposition happens pretty effectively because nitric acid is sticky, so it has a high Henry's law solubility constant. So it means that any surface that has water on it, it'll stick to, which drives a pretty high deposition velocity into forested ecosystems. Another source of nitric acid to these remote atmospheres can come through transport of pans in the free troposphere. So when they descend from the colder upper troposphere, they can decompose to give you back NO2, and then you can undergo this chemistry again, bringing nitric acid into the terrestrial ecosystems. And if you want to be able to sample that quantitatively, way downwind from sources like continental sources in Toronto, the way that you would go about it passively is with a selective material. What we want to do in this case, go to the literature, find out what's been done before. Turns out nylon actually has really great selectivity for sampling nitric acid. This is a polyamide polymer. The amine functional groups on this, we think, nobody's ever shown the mechanism, actually controls the uptake of nitric acid. So you're getting essentially ammonium nitrate forming on the surface, and it's a quantitative collection for nitric acid on that surface. So if we have a nylon filter then, and nitric acid can just diffuse to that surface at a constant rate that only depends on its concentration in the gas phase, we can back out over a month what that average concentration in the gas phase is. we can start to parameterize how much nitric acid might be depositing into those ecosystems. We take our nylon filter, now that is selective, and we try to increase its selectivity further. So we sandwich it between two Teflon rings and protect it with a Teflon filter to keep out things like particulate matter that might deposit on the surface, other debris, any water that might be condensing if there's dew formation going on. And we sandwich this all together inside a petri dish, put it inside some fancy spray painted plumbing parts that we picked up at the hardware store, and create a nice weatherproof shelter in order to get quantitative collection over long time periods. This works really well in polluted environments. There have been literature reports of that before. But the challenge here was to be able to sample these at ultra trace levels, so under 100 parts per trillion by volume. The calibrations that had been done before for the sampling rates as a function of the amount of, or the exposure that you have as a function of the nitrate you collect on a filter had only been done under polluted conditions or simulated polluted conditions. And so our environment that we were going to be sampling in was way down in the bottom corner of this graph. And we said, well, it looks like it's a linear response there, but how do we know for sure? We actually have to go and do the measurements ourselves and validate the whole method. And so this is how we would determine how sensitive that passive sampling approach is. Is the response linear? If it is linear, how much can we collect per unit time? Down to doing good characterization of the samplers themselves. What we actually surprised ourselves with a little bit was that we can use all the different samplers in the literature to come up with a pretty robust description of how these samplers work based on these nylon filters. If you know the diameter of the opening of your sampler and take its ratio to the height from the entrance of the sampler to where your nylon is located, there's a fairly good linear relationship that falls out of that that describes the dose response rate of your particular passive sampler. So we took all the different reports in the literature, came up with linear regression and approximated by calculation what we'd expect here in this blue point. And then we did an active intercomparison where we took our samplers to very low concentration areas, ran them side by side with the typical technique like an annular to neuter, and found that we got the exact same sampling rate out of that. So it looks like we actually have a pretty good description there. So we have a validation of the sampling rate for very low concentration environments. We've confirmed that we're capable of making that measurement very sensitively. So we're out here with a high dose response. So we collect a lot of nitrate at small doses. That means that we could then go on to make quantitative measurements under field conditions. And so this is all Brian's work in terms of the field data, and a master's student who's working with me now as part of our undergraduate project worked up all of that sampling apparatus calculation. And so we have this sensitive and selective method now. We have to get reproducibility. Can we take a bunch of these to the field, put them out for a month, and not contaminate it during our 2,500 kilometer drive out and 2,500 kilometer drive back while we're extracting all that kind of stuff? So Brian put a lot of hard work into this, and what I'm showing here are some IC chromatograms. I'm sure lots of the Murphy group members like seeing these, where we can see the nitrate peak here in our samples, this black peak, and overlaid on top of that are our field blanks and our reagent blanks. So reagent blanks are looking at contamination or extraction solvents, and the field blanks are blank nylon substrates that make the whole trip with us. And the QA/QC protocols that we put into place, which is literally taking stock of every single thing that you touch that you could possibly manage to transfer nitrate from some other surface onto your sampling substrate. We pay very close attention to that, and we effectively have no difference in the nitrate levels in our field blanks compared to our solvents. So we get nice quantitative measurements out of these particular samplers. And what we're able to do then is actually calculate the fluxes of nitric acid into our different ecosystems across that transect. So we do all this nitty-gritty analytical work so we can make some predictions that we hope are at least reasonable at the end of the day. And so we can calculate the fluxes, and what I'm showing here are the fluxes as a function of the different field sites where we collected these samples over 2 years, with Grand Codroy in the south, going to Eagle River in the north. You can see that the concentration of nitric acid that we sampled drops off, which is kind of what we'd expect if you have long-range transport of pollution. As you go further north, more has been deposited in the atmosphere, has been diluted or degraded or whatever, and you would expect to see lower levels. The error bars here tell us how good of a job we're doing analytically versus other things you know about nitric acid that drive its flux. So the deposition velocity, I said earlier, from similar studies in the boreal forest environments is 3 plus or minus 1 centimeter per second. That's the blue error bars on these flux measurements. Our reproducibility in analytical measurements are the black error bars. And so we have a pretty good hold on being able to do good quantitative measurements of nitric acid to drive our flux calculations, and it's within the error that we'd expect from what we do or do not know about the flux deposition velocities of nitric acid into those systems. And so we do this across all of our different passive samplers, or all the dry deposition across NL belt, and we're able to come out with a picture that looks something like this, where we can talk about a little bit more the quantities of reactive nitrogen coming in a variety of different chemical forms across a transect. And so as what we find is that as we go from south through the middle of the transect to the north, that all of the deposition of reactive gases into these forested sites drops off, consistent with long-range transport to those areas, but can't exclude local sources. Most of you will notice that there's a ton of NO here. Acidic forest soils are known to be pretty big NO generators, especially in the boreal forest. So we're sort of decoupling that right now. The seasonal dependence of that is what we're getting at over time. And so we have seasonality in composition that's consistent across NL belt, and it's the source apportionment that we're working on that's ongoing with this. So as we build that forward, we're drawing off of our precipitation data, and this is where our second challenge came in with the NL belt, where we have no power in these sites, and so we had to come up with a way to quantitatively collect nitrogen coming into our remote ecosystems in rainwater, and only collect that rainwater when it's raining. What we did was we built custom off-grid samplers, so we designed all the electronics ourselves, and the sensing application, we did it all in terms of solar power top-ups. So we have battery-operated rain samplers down here. Rachel helped build all of those, so credit there for Rachel. Thank you for helping me build these. In order to capture rain when it's coming in two forms, open fall and through fall. So incident precipitation with no canopy in the way is what we call open fall, and through fall, which is rain coming through the forested canopy, where a variety of different processes can then be decoupled. We collected these in triplicate in both places, so we could have at least some statistical measure of the nitrogen chemistry that was going on in both. We also put out total deposition samplers that collect both dry and wet deposition. So one thing I haven't had a lot of time to talk about, and I won't touch on too much more here today, is the dry deposition of particulate matter into these ecosystems. These samplers catch that deposition input. We were able to then deploy these over a month-long period. So we spent a lot of time optimizing the samplers to be sensitive to low levels of pollutants and rain, making sure they would run for a full month on a battery pack, in some cases 2 months in the most northern site, in order to get good quantitative measurements, and then take them back to the lab and analyze them for pretty much everything that we can think of. So we did total dissolved nitrogen, carbon, phosphorus, and a variety of different elemental quantities that we can measure. We did most of the nutrient measurements because that's what we were concerned with in these forest sites by colorimetric methods or ion chromatography methods. We managed to collect data continuously from 2013 to 2016. What we found was that the wet deposition of reactive nitrogen follows sort of the same trend that we saw in the dry deposition, but it's 10 times greater. And so what I'm showing here is the total nitrogen deposition in our 3-year periods, with the black through the green being the transit from north to south, where you can see the reactive nitrogen deposited in the rainwater increases with any year. But between years, you'll notice that we have almost order of magnitude difference in terms of the variability of the nitrogen that comes in. So Newfoundland sort of sits in a notorious part of the continental outflow, where we may receive a lot of continental outflow in a given year and a lot less in the following year. And so we can have fairly spurious transport events that could drive a lot of this, particularly with wet deposition. So we saw, I don't have time to show today, the seasonality of reactive nitrogen inputs is peaking in the summer when we have lots of rain, but it's also when we have lots of air mass transport from the continent. So the picture that we get by compiling both the dry and wet deposition is one that's more consistent with long-range transport of pollution, but we can't really decouple local sources until we dig into the data in terms of all of that elemental analysis a little bit further. But what we can start to do, and understanding where the pollution comes from is one thing. The major focus of this project was to understand what the impacts are on the carbon storage in the system. So we know what the fluxes into the system are, We've measured those. What are the effects that we could have on carbon storage in soils, ecosystem productivity, and one key that we actually had under our belt from all the measurements that we were making was the measurement of atmospheric phosphorus from wet deposition and dry deposition on top of that. And so if we have reactive nitrogen coming in, we have quite a lot of it for this part of the world anyways. If it coincides with phosphorus, do those two nutrients together create a different story in terms of the potential fertilizing effects that we can see impacting carbon storage and forest productivity than just nitrogen alone? And so these measurements were made by Courtney as part of her undergraduate thesis with a bunch of help from Brian. We measured total soluble reactive phosphorus, and total dissolved phosphorus by two different methods. And a lot of hard work went into making sure that the detection limits for both of those were similar so that we could determine organic phosphorus, which again, I won't touch on today, but it's really to minimize the amount of error that we have in those measurements so we can actually say something meaningful about what's going on. So she did a pile of extensive quality assurance, quality control work, doing standard additions, matrix matching, and making the same measurements with a variety of different instruments to make sure that what we thought we were measuring for phosphorus was in fact the true quantity. So we can say something about the total amount coming in, but also just the inorganic fraction and the organic fraction by difference. What we found for phosphorus was that the amount coming in kilograms per hectare per year showed that same gradient popping out with low amounts going in the north and the high amounts going in the south, but now we started to see something different. So these samples we broke down by wet deposition versus dry deposition, similar to what we did for the nitrogen, but in this case we see that dry deposition is actually what's driving the difference in the gradient. What we know about atmospheric phosphorus is that it comes from a variety of aerosol sources. So desert dust tends to be the major driver on a global scale. And so we're still trying to understand where it is coming from, but we see dry deposition affecting inputs on a Transect scale with greater amounts coming in the south. And the amount that's coming in the wet deposition, which I'm showing here in gray bars, is fairly consistent between all of the sites. So the dry deposition seems to be really driving that. And in order to try and understand where this might be coming from, it could be desert dust, it could just be internal cycling of pollen in these sites. The temporal nature doesn't match up with the latter explanation. So we're still trying to figure out what the atmospheric source is. If you go to the literature, the estimates that you see from global models, our measurements line up with those very well. Where the quantities we're seeing in the different regions don't match up perfectly, like we might expect, but the sources in that model are highly uncertain. If we look at the pollen sources, different fractions of plant material or desert dust, the uncertainty in the deposition, particularly in this part of the world, but across most of North America, spans about a factor of 1,000. And so the uncertainties in the model, the fact that we get a match here at all is probably completely circumstantial. But we've been able to make the measurements, and this is what they are. So hopefully we'll start to understand that a little bit more. Having those two measurements now allows us to say something a little bit more meaningful about the growth and productivity of the forests. So we looked at the total dissolved phosphorus and total dissolved nitrogen relationship. We looked at the ratio of this because what biogeochemists will use is the nitrogen to phosphorus ratio in plant litter or in soils to understand effectively what the nutrient limitation in those environments are. And we looked at this from what was coming in the wet deposition or total deposition in our precip samples. What we found was that there's a modest correlation between the two. So the temporal nature is modestly correlated, and that nitrogen to phosphorus ratios across NL belt were lower than in litter. What this tells us is that the nitrogen and phosphorus coming in at the same time could have a fertilizing effect. So it's enriched in both of those nutrients compared to what is coming in from the needles and leaves of the trees dropped to the forest floor. That simultaneous deposition then could stimulate growth. Something that we found in terms of the biogeochemical budgets is that the nitrogen coming in is equal to the same amount that comes in from the litter on a 4 to 10 year time scale, depending on whether you're looking at the most southern side or the most northern side. So the nitrogen that's coming in from the atmosphere is significant compared to what comes in from the leaf litter. So we are increasing the nitrogen in those environments in that way from atmospheric deposition. Meanwhile, the phosphorus that's coming in is really quite small compared to the litter fall phosphorus. That doesn't mean that the phosphorus coming in from the atmosphere isn't important. It's all about speciation, and that's sort of the direction that we're going in now. So we know that nitrogen inputs and availability in the southern region are greater. There's a higher carbon turnover. When you have high carbon turnover in forested environments, it increases nitrogen availability and will stimulate forest growth. So adding more nitrogen there could stimulate the growth further. but making that phosphorus available from the atmosphere may actually be what's helping that forest ecosystem absorb even more of that nitrogen. And so it coming in the form of phosphate, which is the dominant form that we see in our dry deposition of phosphorus, that might actually be more bioavailable than the phosphorus coming in from the litter fall, and that might drive some further growth in those forests. And so that sort of wraps up this section of my talk. So hopefully I've convinced you that a lot of nitty-gritty, painstaking analytical and instrumental work can let us make alter trace determinations of species in remote environments. And we can do this without using any power or by being creative and coming up with unpowered solutions that will operate remotely in these types of situations. We're able to do when we applied those sort of principles is quantify dry and wet deposition of reactive nitrogen across latitudinal transect, where we saw reactive nitrogen decreasing with increasing latitude. We were able then to determine that dry deposition represents like 10% of that input, while wet deposition dominates the inputs. When we take the additional measurements of soluble reactive phosphorus, we're then able to say a little bit more about the fate of soil organic matter and the productivity of the forest on top of that. And then the last thing I just want to reemphasize here is that the sources of soluble reactive phosphorus and reactive nitrogen in these different samples are still kind of up in the air. We've been doing some comparisons with an ex-Murphy Group alumni, Jeff Geddes, in nitrogen deposition driven by geochem models. The deposition quantities are similar, but the speciation is different. So we're working together to try and figure out what might be going on there. And then the soluble reactive phosphorus, like I indicated earlier, there's a lot of uncertainty there. So there's a lot of work in terms of understanding where atmospheric phosphorus might come from in that part of the world and drive the deposition to understand all that chemistry. One last thing here, again, this is for Rachel. The samplers work so well that we have made a second generation of them, and they are going on to do more work up at York to this day. So they continue to exist, and they're still out there. So this project was also quantifying ammonia. So you guys might have seen some dry deposition of ammonia calculations for passive samplings in the previous slides. And this is sort of my segue. It's a bad segue into talking about how I got into indoor chemistry, but it's really derived from making some more of these passive gas sampling measurements. So we were trying to understand the quantities of ammonia across the NL belt. And simultaneously, I was also still fascinated with amines. And so I figured if we're trying to quantify ammonia in these ecosystems, why don't we try to get amines as well? They're really challenging to quantify. They're in the atmosphere. They're at perfect quadrillion levels, which makes them really difficult to detect. They break down really easily by oxidizing with ozone and OH, but they can really readily react with sulfuric acid or nitric acid to form new particles in the atmosphere. And that has a variety of other concerns. We'll go into that in detail, but the outdoor sources of these amines are poorly constrained, mainly because they are present at very low levels, which means that we don't really know exactly where they come from. Their temporal nature is a little bit elusive. Identifying sources that aren't enormous sources is challenging. And so it's important, there's important chemistry here to get our thumbs on in terms of understanding how the structures and sources of different amines are driving this type of chemistry, but it's difficult to discern because they're hard to detect and we aren't very capable of speciating them. And so this, like I said, was pursued further as part of the biogeochemistry work at NL Belt. And it was building off of a frustration of mine for my PhD, actually, with Jen, where we could never fully resolve trimethylamine and diethylamine by ion chromatography. And I was continuously frustrated by that, but luckily there were some people who hadn't experienced the frustrations of trying to separate things by ion chromatography yet. Brian and a summer student, Leah Gulte, who's doing her master's at UPEI now, decided that they were going to try and deal with this coelution issue, and they actually pursued some good ideas. We looked at increasing the temperature of our columns to resolve those compounds, and we actually found that effectively separated them. So this is the coeluting pair here, 4 and 5. As we ramp up the temperature of the columns, we got full separation between those two. Little did I know that this was bringing me into the wide world of indoor amines and their quantitation. The reason why this turns out to be a good approach for something like that is that the temperature control didn't just give us the ability to resolve those two coeluting amines. It gave us the ability to resolve the 11 most abundant atmospheric amines that have been reported both indoors and outdoors from the full suite of inorganic cations that we'd expect to find in atmospheric samples, whether it's particulate matter or the gas phase. So it's monoamines, diamines, and this temperature selectivity it actually mimics organic solvent addition to your mobile phase. So if you have IC components, especially typical ICs now, they're not compatible with organic solvents. And so this temperature programming actually allows us to get the same selectivity effect without having to deal with handling organic solvent inside those systems. And it overcomes a major limitation if we're using direct mass spec methods. So if we're using a SIMS method or an aerosol mass spec method, you can't tell the difference between structural isomers. A C3 amine looks like a C3 amine, a C2 amine looks like a C2 amine. And this separation method actually can resolve all the sets of structural isomers that we'd expect within this suite of 11 atmospheric amines. And so we had a great method from first principles. We had to test it under real atmospheric worst case conditions. So in collaboration with Cora Young and her PhD student Rob DeLorenzo, again, both U of T alumni, out in Newfoundland, we had pretty much the worst case atmospheric sample I could think of. Biomass burning samples from forest fires that occurred in Quebec and Labrador in 2013. Got transported to St. John's, but only after spending a long time in the coastal zone, picking up tons of sea spray. And so we had these biomass burning particles that were mixed with sea spray, fully loaded with things like potassium, sodium, and ammonium that can mess up an amine measurement pretty easily. So we want to know, are amines there and can they be quantified? Short answer is yes. If you want to learn more about the chemistry that's going on in all that, all those samples, you can look at Rob's paper from last year in ACS Earth Space Chem. We can separate our analytes from the matrix, no problem. So for the students in the room, if we want to separate and quantify something by chromatography, we have to have a resolution of at least 1. This method is capable of doing that, even when the ratios of our cations to our target analytes in the form of amines are present at ratios of 1,000 to 1. So we figured we had this method, works on these really dirty samples. We can probably apply them to our anal belt samples, but we didn't know that there was a similar need in the indoor environment. So this is my long and poorly constructed segue into talking about indoor chemistry. Amines in indoor chemistry have totally different implications. It's because their atmospheric fate gets changed indoors. So you can have amines emitted from a variety of different things: cooking, smoking, textiles, decomposing food, other decomposing organic matter, or even your favorite kitty cat might be making amines indoors. They can then go on to react with other compounds that are in abundance indoors that aren't in abundance outdoors. So things like HONO, we know are in abundance indoors from work from John's group, some work that we've done recently I'll talk about today, to form nitrosamines. They can form in the gas or condensed phases, and these are known carcinogens. So you can have exposure by the chemical reaction product of amines and HONO. So two things I like a lot coming together to make a terrible situation for an analytical chemist indoors. And so the indoor photolysis and photochemical oxidation of amines and nitrosamines indoors are shut off because there's not a lot of light. And we're starting to learn a little bit more about OH indoors, but generally speaking, we don't think that this would be a dominant fate of amines indoors. And so their fate is likely controlled by equilibrium partitioning to surfaces or to aerosols, or reactive transformations like the formation of nitrosamines, which then also undergo the same equilibrium partitioning processes. And so at the end of the day, to understand which nitrosamines might form, we have to know which amines are there in the first place, and then understand their chemical reactivity from there. So this method that we came up with that can speciate and quantify all this wide variety of amines, the most abundant amines that we might expect to find in an indoor environment, would allow us to address this potential risk factor, the formation of nitrosamines indoors. And so this is sort of the goal of the Sloan Foundation, who I'm currently funded through, is to understand this type of chemistry that could happen indoors. Where do amines come from? How much? What are they? How much of them are there? And what chemistry can they do? This is because we spend more than 90% of our time indoors. You live to be the ripe old age of 90. That means you spent 81 years of your life in a room like this one, or your bed, or whatever. And you're exposed to the atmosphere in these spaces through that whole period of time. And so if there's detrimental impacts from being in these environments, either through acute or chronic exposures, driven by the chemistry that happens in these spaces or the emissions that happen in these spaces, it's very important for us to know. Basically, the chemistry of the indoor atmosphere is largely unregulated outside of what makes us comfortable: temperature, humidity, in CO2, and things that kill us pretty quickly, like carbon monoxide. So meaningful measurements in these environments then also require the fact that we use them the way that we normally would. So to go into an unoccupied environment and make measurements there is one thing. You're not disturbing people. You can bring in all the mass spec pumps that you want. But if you want to actually get at what's going on in people's day-to-day lives, you have to have them using the space normally. And so what I've been getting into now is developing safe and widely available instrumentation that can come into these environments that disturb that use of the space as minimally as possible, that also give us information like the quantities and species of amines in those places, so that we can then get out and do surveys through hundreds or thousands of homes to get a handle on the chemistry that might be happening, but also the variability we might expect in different indoor environments. And so coming back to amines, this is pretty fortuitous. The conference in Poland put on by the Sloan Foundation really drove me to try and make these measurements in our own house in Newfoundland, test house number 1, maybe I'll call it, in test house number 1, where there are 2 vegetarians by and large, and food and degradation of organic matter is one of the best known sources outside of cigarette smoking in terms of emitting amines into an indoor environment. And so cooking and food prep processes were deliberately targeted over a 1-week period, looking at different vegetarian things, and the one opportunistic carnivore had the opportunity to cook up some known amine sources like fish cakes and salmon to see if common amines that had been detected from degradation of this or the cooking of this type of stuff could be picked up by passive sampling in the indoor environment. And so the passive samplers are the same ones that were used for the outdoor environment to collect ammonia. So the same passive reactive samplers worked really well for ammonia. All of our samples that were collected either on an event basis or integrated over time showed an increase in ammonia as we increase the sampling period, which is what we'd expect with this type of sampler. So you want your product to be stable on it. The integrated mixing ratios were actually quite a lot smaller than we might expect based on literature. We only saw somewhere between 5 and 63 ppb in the indoor environment during this time. We saw all kinds of amines. I wish I could say that they were actually there. None of them were above the detection limits. They were in the blanks as well, and we can't really say that all the ones that we saw were there. They came in the background as well. So it seems like these samplers are capable of finding them, but only trimethylamine and monopropylamine were present in a way that we could say we definitively saw them. And trimethylamine is the only one that demonstrated a pattern of accumulation over time, similar to ammonia, that tells us that it's being sampled effectively, but also that the product is stable over time. We saw monopropylamine in 5 different samples. Their levels were exceeding the negative controls, but we didn't see any accumulation being observed. So these are event-based samples here, and we start to see sporadic presence of this above our detection limit in a shorter period sample, so an integrated sample here across all of our cooking events, and then a 1-week integrated sample, and that is showing a decrease over time. I would suggest maybe the product on these reactive pads isn't stable. So this sort of got me involved in indoor chemistry. We found that amines are ubiquitous, but the passive sampling strategies that we have available to us right now, they're not quantitative. What I'm showing on the y-axis here is the target analyte quantity in micrograms. We know the sampling rate for ammonia, and we can back out a mixing ratio. We don't have these parameters for amines, and we don't even know if they're stable products on the surfaces. This is part of the research that I'm currently doing. Layla has transitioned to working on this as part of her master's research. So this means that the duration that we have to collect passive samples by this type of strategy in the indoor environment in order to get over our detection limits remains unknown at this point. I'm just going to quickly talk about a couple other projects I was involved in because I'm really excited about these from indoor chemistry, also because Arthur wanted to hear about some of the work that I've been involved in when he invited me to talk today. So this is for you, Arthur.
Thank you. Everyone else can go up.
So a couple other projects I've been involved in are based off of my love for Hono. And so I've gotten involved with a couple other research groups to try and tackle measuring Hono in indoor environments, but doing it in ways that we don't need to bring in a $500,000 mass spec to make that measurement. So we came up with this instrument in collaboration with Tara Kahan and her postdoc Shan Zhu to modify Knoxbox to make measurements of Hono. So outdoors, Hono is a known quantitative interference in a NOx measurement, but outdoors there's not a lot of HONO. Indoors is a different story. So you have this quantitative chemical interference when you measure reactive nitrogen, so NOx, NO and NO2, indoors using chemiluminescence detection that would be biased by HONO being present. But if you put a sodium carbonate denuder in line with your inlet and turn that on and off, you get HONO by difference by scrubbing it out. Past measurements tell us that there's not a lot of nitric acid indoors, and so by difference, we're assuming that everything is HONO. It could contain a little bit of nitric acid as well. We put this instrument together. It's designed to measure oxidants indoors. We call it the mobile instrument for something oxidative species. There's an L in there. It's called Milosh, which is another Murphy group throwback. What we found, though, was that the indoors, similar to some studies that were done here in Toronto, had a gas stove in it, and there were huge NOx quantities that were produced, as well as direct emissions of HONO. And we were using this instrument then to look at oxidation chemistry going on indoors. So here's just a snippet from the paper where we're showing the concentration of NO that we observed, and on the right axis here is NO2 as well as HONO being plotted in parts per billion, which you can't see. But every time you turn on that stove, you get these huge pulses of NOx out of it. That includes direct emissions of HONO, and the levels are enormous. When I saw this, I really started to get quite concerned. We saw levels exceeding 10 ppb regularly. This event here, you can't really see it's the green line for HONO, is up around 40 ppb at the top of that particular event. The range hood is on. That has very, very large concerns for me being inside. So direct exposure to HONO is bad in the first place. The second thing that we realized, we went to the literature and said, why has nobody ever measured this absurd amounts of HONO indoors before? Well, there's some measurements that say that there are large levels, but it's not really this high time resolution measurement. So being able to see this like really strong episodic emission isn't really out there. There are a few measurements that have been made that are quite nice, but most of what's out there are NO2 measurements that were quite high, but it turns out that most of them are made by passive sampling, and all the passive sampling methods take NO2, convert it to nitrite, and then you analyze for the nitrite. So if HONO goes onto those surfaces, this is a proton, you just have nitrite left behind. And so passive sampling determinations of NO2 in the past, we concluded were biased high. And the episodic component here from cooking, we're still not quite sure what to make of that, because people have deliberately tried to make HONO measurements in the past, and they've seen some large levels, but by and large, the literature reports pretty low HONO mixing ratios in indoor environments. So the background levels that we saw were similar to that, but during cooking episodes, these episodic periods, the NO2 and HONO were really quite high. And that was quite surprising. So the instrument that we built was able to do this, and the whole goal of this study, which is driven by Tara, I'm just the person who helps you measure HONO, was to understand the oxidation chemistry that could come on from this. They used a photometer to go and look at the solar irradiance, not solar radiance, just irradiance in the indoor environment. They found that sunlit portions of the residence with this much HONO in them can make OH at background levels on the order of 8.3 times 10 to the 6 molecules per cubic centimeter per second. And during those cooking events, you have that HONO mixed into a sunbeam, and you're making 97 or 9.7 times 10 to the 7 molecules per cubic centimeter per second. And so this could drive a lot of OH chemistry indoors, which, if you remember back to my original slide on amines, we don't think there's a lot in indoor environments, but this new work that we're doing suggests that it could be actually possible to have HONO driving some OH production indoors. And so this is one little snippet of some HONO work. So there's a big HONO problem indoors. We should work on that. And here's one potential solution. So this is a collaboration that I got involved in while I was at Boreal with metal organic framework chemist Mike Katz, and in collaboration with Corey Young as well. So we call him the Moth Father. This is something he likes to be called. So how these work, you have metal nodes that you create a porous network with by bridging them with organic linkers. You can create very well structured pores that can be hexagonal, bipyramidal, or tetrahedral, or you can stack them together into these highly porous crystalline substances. Those organic linkers, if you chemically modify them, can then allow you to control some chemistry that can happen within the pores of those. So you have huge porosity within these. I'll refer you to the paper. Hopefully it'll be through review soon. The revision was just put in, it should be done in the near future. They have huge surface areas internally that you could just sorb gases onto, but if you do chemistry, you can actually come up with a much better solution. And so the chemistry that we got into looks to convert HONO to N2 and water. And we've been able to do this and show that we can do this within a metal organic framework. So what we do is we produce a known quantity of HONO using a typical HONO generation system, doing acid displacement of sodium nitrite with hydrochloric acid. We then take a very tiny sample of MOF. We put it in a 1/4-inch tube. It's like this long. Sometimes we put a whole gram in there, and then we expose it to HONO for long periods of time. We measure it, again, using a modified nitrogen oxide analyzer to look at the HONO that's getting through that MOF as a function of time. When we take a metal organic framework that's based off of zirconium oxides linked with these particular linkers here with the hydroxyl on them, we can see that HONO will be sorbed onto the surface. So this is the concentration over the initial concentration of HONO. Then we modulate the MOF in line, and eventually the HONO will break through that MOF. Basically, all the surface sorption sites have been occupied, and the capacity for the MOF to retain HONO has been completely occupied. You turn off the HONO, you can then desorb it back out. So you can have a temporary storage of HONO inside of the metal organic framework, and you can turn it back on and off. So sorb and desorb reproducibly to sequester HONO, and then maybe take your MOF somewhere else and then flush it out is one approach. But if you do some chemistry where you put an amine group on this particular linker, you can actually chemically scrub HONO from that gas stream. And so here I'm showing that same normalized concentration as a function of exposure time. So MOF chemists like to use this normalized time in days per gram of MOF. So days of exposure at whatever concentration per gram of MOF. I'll just draw your attention to the fact that we went up to 100 here and did 3 sorption-desorption cycles to a complete breakthrough down to a complete desorption. Here we're at 1400, and we don't have any HONO coming through our metal organic framework with the same amount put into that tube that we're trying to break through on. In order to see anything coming through it, the chemistry is very effective at scrubbing HONO. We had to start to dilute it with the unfunctionalized version in order to actually see the consumption of all the amine functional groups in it. So we have so much porosity, we have so many organic functional groups to do the reactivity within this metal organic framework that's very, very selective for scrubbing HONO. When we dilute that, we can then start to see a HONO break through. Again, this is over the same period of time. So these experiments take about 90 days to do. So doing this full breakthrough, it was going to take us years. It took us 3 years to do this project anyways. So we didn't want to extend it any further. But you can see that eventually, if we integrate all the HONO that gets sucked up over this period of time, that amine group is getting quantitatively used in the conversion of HONO. We've gone further with this and actually figured out what the mechanism is. So this is what we originally designed the metal organic framework for, is to perform this heterogeneous Greet-Salzman reaction. So anybody who knows about the low PAT for measuring HONO or for measuring nitrite by chemical derivatization, you convert it to a diazonium dye. Basically, we're doing the same thing, except now we're doing it heterogeneously. So you take HONO from the gas phase, we react it on the surface of the metal organic framework that's functionalized with this amine group. This zirconium oxide node actually allows us to form a nitrosonium cation on the surface by proton exchange. We lose a water here, and then we lose another water as the nitrosonium cation reacts with the amine functional group to give us the diazonium product on the surface of that linker, which then acts as a leaving group in the presence of water, releasing nitrogen, giving us the products of water and N2 from HONO coming in the first place. So we've completely deactivated HONO to N2 through this heterogeneous conversion inside a metal organic framework. And all we need is a little bit of water to get to our end product here with the hydroxyl group. So we've gone through most of this mechanism. We're pretty confident that this is what's going on. We can see a lot of these intermediates by different spectroscopic methods, powder X-ray diffraction, and looking at the changes in surface area over time. So it seems pretty effective at essentially denoxifying, not really dehonoifying, an exposure stream of gas. Just to give you an idea of what that is from this particular plot again, loaded about 1 gram of UIS 66 that's aiming functionalize, exposed it to 60 ppb of HONO and 100 cc's of gas flow, and we had over 100 days of effective removal and saw no signs of breakthrough. So this high porosity material might be a very good method moving forward to scale up decontamination of HONO in the indoor environment where we have these very high levels. So it might be something that could be really nice to do. And so some conclusions up there, some upcoming events for indoor reactive nitrogen here in 2019. See Miriam's in the room. We're involved with Health Canada doing new home air quality study. So that's getting going tomorrow, I think. Samples were sent out on short notice yesterday. So that's going to start happening. So we're going to be going into a lot of new built homes to look at indoor chemistry there for reactive nitrogen. We're also going to be going back to Syracuse to work in our collaborator Jen Zhang's indoor controlled HVAC room where we can create our own built environment and perturb the chemistry there as we see fit to understand more about what's going on, but also to test all these newly developed instruments and methods. So I'm just going to shamelessly plug a couple of last things here. Next week, we're only connected by a subway now, guys. If any of you are interested in learning about how Picaros are used, we're doing a Picaro user science meeting next week, next Friday. We have a variety of guest speakers. If you want to come, please RSVP. There's free coffee and food, and you can learn some stuff about what using these cavity ring down spectrometers is all about for HCl, CO2, methane, all kinds of stuff from a variety of scientists around the GTA. So if you're interested, please RSVP or email me. One last thing, myself and Corey Young are hosting a session at CTAC this year, which is in Toronto. So graduate students that are in the room that might be working on indoor chemistry, we have a session just for you, chemistry and exposure in the indoor environment. You can submit your topics now, so abstracts are welcome anytime now until June 5th. So I encourage you all to join us. All kinds of topics involving indoor environments, so chemistry and exposure are very welcome. So we'd be happy to have you all come there. And so again, couldn't do all this without all the fantastic people who drive the science, as well as you guys for being here today. So thank you so much for your time and attention. Take any questions.